Bueno,
It's a great pleasure for me to introduce Oriol Dols-Icardo. Oriol is a postdoc at Hospital Sant Pau. As you know, Hospital Sant Pau, I think, is the most relevant unit in the field of dementia, and Oriol genetics there. I was lucky to be in his doctoral panel in 2014.
16.
16, okay, almost 10 years ago.
Sí.
And at that time, he was playing a very, very important role in the HESCO. As you know, is the consortium of genetics, and his, his PI at that time, Jordi Clarimont, was really a tractor in the HESCO, even though Agustin and myself were the founders. But I have to admit that Jordi, and together with Oriol, they played a fundamental role at the beginning, and opening the HESCO to other phenotypes. That Oriol has been doing this since that moment. He has been working in ALS and frontotemporal dementia, and working in sequence. We were working in more primitive techniques, and he started working on sequencing, and today he has continued with this phenotype with more advanced technologies, and he's going to talk about his new final study. So thank you very much, and it's a pleasure.
Thank you for the presentation, Pascual, and thanks for inviting me to give this presentation and share our research. So, first of all, I will briefly introduce ALS and the concept of neuroinflammation. Then I will continue by discussing the role of the peripheral immune system in neurodegenerative diseases and in the context of neurodegeneration, but also in neuroinflammation. Next, I will move to discuss and link the evidence linking the peripheral blood immune cells with ALS, not only in the human blood, but also in the human central nervous system. And finally, we'll conclude this introduction by highlighting the main advantages of single-cell RNA sequencing over flow cytometry. So next, explain the main results of the manuscript we are going to submit to Journal of Neuroinflammation this week. And finally, I will show the research lines I have, which are focused not only in ALS, but also in other neurodegenerative diseases in the context of the peripheral immune system. So amyotrophic lateral sclerosis is characterized by the degeneration of upper motor neurons in the motor cortex and lower motor neurons in the spinal cord. At postmortem evaluation, more than 95% of cases show phospho-TDP-43 aggregates. But another major hallmark of ALS and neurodegenerative diseases is neuroinflammation, which is characterized by the expansion of microglia and astrocytes, which acquire a reactive state. But cumulative evidences show that the peripheral immune system, peripheral blood immune cells, infiltrate into the brain, contribute to neuroinflammation and neurodegeneration, and are known to be a major hallmark also, not only in ALS, but also in other neurodegenerative diseases. So for example, so since early in the 90s, we know that peripheral immune cells infiltrate the motor cortex and the spinal cord of ALS cases. So here we have in ANC examples of CD4 and CD8 positive T cells. These are lymphocytes, CD4 positive and CD8 positives, and that they are located away from capillaries and blood vessels. In other neurodegenerative diseases, as I just mentioned, they also play a major role. So in Alzheimer's disease, we know that CD8 positive T cells patrol are expanded in the CSF of Alzheimer's disease patients, but also that CD8 positive T cells are accumulated around A beta plaques. We also know that T cell infiltration correlates with Bragg stages, but with tau pathology, and that this is independent of the beta plaque burden in the brain. We also know other things, such as T cell, the blocking or depleting T cells exacerbates tau pathology. So we have different relationships between T cells and Alzheimer's and Alzheimer pathology. By the other hand, in DLV, we know that CD4 positive T cells are also increased in the CSF of these patients, and that these cells, CD4 positive T cells, are accumulated around alpha-synuclein and alpha-phosphosynuclein. While in FTLD, in Pick's disease and PSP, so in FTLD tau, we also know that CD4 and CD8 positive T cells infiltrate the brain. So returning to ALS, we beyond CD4 and CD8 positive T cells, we know that natural killer cells accumulate in the spinal cord and motor cortex of ALS patients. But as it happens in other neurodegenerative diseases, such as Alzheimer's disease, DLB, or FTLD, we also know that CD4 positive T cells and CD8 positive T cells also infiltrate the central nervous system. In the blood of ALS patients, using flow cytometry, we know that, well, several reports have demonstrated the involvement of CD8, CD4 positive T cells, monocytes, and natural killer cells. But the problem is that not all these results have been replicated in all studies. So some studies report monocytes, other studies report that natural killers are increased, other studies report that CD8 or CD4 positive T cells are expanded in the blood of ALS patients. So the point here is that not all studies replicate the same results, and that may depend on the stage of the patient, on the age of inclusion of the patients, on the genetic background of the patient. Several factors may influence this outcome. As I mentioned before, these studies use flow cytometry, and which are the limitations of flow cytometry? So some of the limitations of flow cytometry is that they rely on a preselected list of markers. So they rely on the selection of antibodies. So they use a hypothesis-driven approach. So in case that we want to determine the proportion of monocytes or isolate monocytes, we must use CD3, CD14, and CD16 to classify these monocytes into the two main populations. Or in natural killer cells, we need these antibodies. So we need to select some antibodies to finally determine the proportion or to isolate these cells. So this is not an unbiased approach, and also it depends on the combination of fluorophores. So as you may imagine, the number of detectors and the number of channels is limited. So we cannot use 100 antibodies in the same approach, in the same experiment. So as a consequence of flow of this technique, of this biased technique, flow cytometry, the researchers have traditionally classified into two main populations, the natural killer cells. So in this case, we see here the CD56 bright natural killer cells, which represent 10% of the total natural killer population. From now to the end of the presentation, CD56 bright natural killer cells will be bright natural killer cells. And then the other part, the main population of natural killer cells are the ones called CD56 dim natural killer cells, which represent almost 90% of the natural killer population. And they are characterized by the high expression of CD56. Natural killer bright cells have a high expression of CD56. And by the other hand, dim natural killer cells have a low expression of CD56 and a high expression of CD16. Importantly, CD56 dim natural killer cells present a more mature and more cytotoxic profile than bright natural killer cells. So in contrast, single-cell RNA sequencing is an unbiased and high-throughput technology, which does not rely on a list of predefined markers. So we don't have to select antibodies. So this is an unbiased technique. And importantly, it gives us the opportunity to feasibly determine determine the whole landscape, the entire landscape of cell populations. So in this case, this is the procedure to isolate peripheral blood mononuclear cells, which are the same, almost the same as peripheral immune cells, blood immune cells. But when we have here, after cryopreservation, we sequence them. So we have a mix of cells. We don't know which proportion of cells we have. We encapsulate these cells into droplets, so we have thousands of cells per sample, depending on your approach. In our approach, we decided to include around 5,000 cells per individual. So these encapsulated cells are then sequenced. So the fact is that the final result we have is that for each cell, we have their RNA content. So we sequence these cells in a cell-by-cell basis, and then at the end, we have an RNA profile for each of the cells. So based on the RNA content or the RNA profile, we can compare different cell types. We can infer, in fact, different cell subpopulations, not only major populations, but also subpopulations. And importantly, this is not biased to the use of antibodies. So as I mentioned before, over the years, we know that bright natural killer cells and dim natural killer cells are the major populations of natural killer cells. But using single-cell RNA sequencing, not only us, but also other approaches in the last year, have identified other subpopulations belonging to the dim natural killer cell population. So the basics of single-cell RNA sequencing is that we sequence the RNA on a cell-by-cell basis, so we know the RNA content of each cell, of the thousands of cells per individual. So beyond identifying major populations and subpopulations, we're able, so it gives us the opportunity to compare gene expression between healthy controls or different groups. So at cell type resolution, so here in a cell type, here in another cell type, so we can also not only determine the proportion of cell types and cell subtypes, but also we can perform differential gene expression analysis. Another advantage of single-cell RNA sequencing is that we can infer cell-cell communication patterns, and then we can compare the cell-cell communication patterns between two groups and identify which cell communication patterns emerge in response to disease condition. So, given that peripheral immune cells play a major role in neurodegenerative diseases, they contribute to neuroinflammation, to neuroregeneration, they are associated with protein aggregates, but also that the proportion of immune cells, of peripheral blood immune cells, have been shown to be altered in Alzheimer, in amyotrophic lateral sclerosis using flow cytometry, and that single cell, so the role of peripheral immune cells has yet to be investigated in ALS using single-cell RNA sequencing. Then we decided to perform single-cell RNA sequencing in a group of 14 ALS patients and 14 healthy controls. Importantly, these healthy controls were cognitively unimpaired. They had a normal MRI. They have a negative AD biomarker profile and normal neurofilament light chain levels. No differences in gender or age at sample at blood collection were found when we compare both groups. And the approach was the one I mentioned before. So we isolated PBMCs using fecal gradient centrifugation. So we layer the blood up the fecal layer. So we centrifuge this mix, and we finally obtain this disc here, the gray zone, the gray region here, which correspond to peripheral blood mononuclear cells, which after a couple of washes, we cryopreserve. So after cryopreservation, we perform library preparation, and we sequence with NovaSeq 6000. So after a strict quality control, I'm not gonna deep into these details because I guess that most of you are not familiar with bioinformatics. So after a strict quality control, almost 110,000 high quality cells remain from these 14 ALS patients and 14 healthy controls. Then our first step was to perform supervised clustering using a multimodal reference PBMC dataset. PBMC is peripheral blood mononuclear cells, which is the same as peripheral blood immune cells, almost the same. I will show you that that's not not, that's not correct. So we have PBMCs, we used supervised clustering, then we identified the major populations and the minor or the subpopulations of cells. But as the second step, we compare cell type proportions and cell subtype proportion using Propeller. We estimated differential gene expression alterations using MAST. We also inferred and compared cell-cell communication patterns between ALS and healthy controls. And finally, additionally, we explore whether the combination of the proportions of peripheral immune cells may help explain the variation in plasma NFL concentrations as a surrogate biomarker of neurodegeneration. So in our first approach, we identified the major population. So we identified 25 major cell types, which correspond to, for example, natural killer bright and dim cells, but also proliferating, and CD4, CD8 positive T cells, which are subdivided into effector memory and central memory, naive, and the same for CD4 positive T cells. So this is the first layer. We have 25 major cell types, and then our first approach was to compare cell type proportions. Then we identified that CD56 dim natural killer cells are increased in ALS, and also classical monocyte or CD14 monocytes, which is the same, and CD56 bright natural killer cells. But this comparison did not reach statistical significance after multiple comparisons, so after correction for multiple comparisons. So in the first approach, in the first layer, from among these 25 major cell types, we found that these 3 cell types are increased in ALS, but only dim natural killer cells are significant after correction for multiple comparisons. So leveraging the unbiased nature and the high resolution nature of our data set, we also identified minor subpopulations. So this is one of the advantages of single-cell RNA sequencing over flow cytometry. So we identified a total of 55 cell subtypes. So we were able not only to identify the ones I mentioned before, but also subpopulations. This is just a small example. Subpopulations of effector memory, central memory CD8 and CD4 T cells in blue and green, or the ones I mentioned before, these 4 subpopulations of dim natural killer cells, NK1, 2, 3, and 4. So now we compare the proportions of these 55 cell subtypes between ALS and healthy controls, and we found that the ones that, again, were increased in the blood of ALS patients and were statistical significant after correction for multiple comparisons were NK2 and NK4. And as you can see here, the most common population is NK2, which represents around 5 to 15% of the total number of PBMCs, and is a major population of these dim natural killer cells. So NK2 is the most common population of these dim natural killer cells, and is the one, in fact, driving the expansion of natural killer cells in ALS. So as you can see here, the fold change is 3 here, and the fold change for the major population dim natural killer cells is 2, and the p-value is also more significant when we compare this subpopulation. So highlighting the importance of this natural killer subpopulation NK2, but also we found that NK4 are increased in the blood of ALS patients, now representing a minor population of these CD56 natural killer cells. Again, also we found that CD14 monocytes or classical monocytes and CD56 bright natural killer cells are increased in the blood of ALS patients, but again, they did not reach statistical significance after correction for multiple comparisons. So our next objective was to compare gene expression between ALS and healthy controls at single cell resolution. So we found around 200 genes deregulated in ALS as compared to healthy controls at single cell resolution. I am not gonna go cell by cell, so I'm gonna focus on a couple of cell types, probably the most important, the most interesting, or the most relevant cell type. So here we have the NK2 subtype, the one that is driving the increase of CD56 in natural killer cells. And we found that the genes deregulated when we performed gene ontology enrichment analysis pointed towards the activation of immune response and lymphocyte proliferation. So the genes that are deregulated in this natural killer subtype are have a function related to the activation of immune response and the proliferation of lymphocytes. And by the other hand, we also, I mentioned before, we have several cell types where we found differential gene expression at single cell resolution, but I'm not going to go cell by cell. So another cell type, interesting cell type, is CD8 effector memory T cells. In this case, I'm showing just the number 5 subpopulation. So the point here is that the genes deregulated in this effector memory T cells point to the enhanced antigen presentation by MHC2 pathway. So you see here HLA, which belongs to DP and DPA, and DR, which belongs to the MHC2 pathway, also C74. So this points that towards the idea that these lymphocytes are presenting antigens in an exacerbated way as compared to healthy controls. So our next approach was to assess was to infer cell-cell communication patterns and assess whether they differ between healthy controls and ALS patients. So here we have two scatter plots. In the y-axis, we see the incoming interaction strength. So is the proportion of single signals that are received by these 55 immune cell subtypes. And in the x-axis, we have the outgoing interaction strength. So this is the signals sent by the proportion of signals sent by these 55 immune cell subtypes. And this scatter plot is split by ALS and healthy controls. So as you can see here, again, NK2 cells, if you focus in these circles, the red circles, you see that NK2 cells receive 3 times more signals in ALS than those from healthy controls, pointing towards the hyperactivation of these natural killer cells, which are receiving a lot of signals, 3 times more, in fact, than those of healthy controls. So we next focus on which is the pathway which is driving the increased signal reception by natural killer 2 cells. So we then compare the signal reception between healthy controls and ALS patients, and we found that MHC1 pathways are increased and are driving the increased interaction strength, the increased signal reception by NK2 cells. So a more detailed examination of the MHC pathway involves the ligand interaction. So the ligand receptor pair interactions here. So MHC pathways include the interaction of different HLA proteins, A, C, E, F, also others, but this interaction is mediated by receptors in other cell types. So we here have in the x-axis the cell types that are sending signals to NK2 cells. So CD4 T cells, CD4 central memory, CD4 central memory 2, 3, 4, and CD8 effector memory T cells, the ones that have an enhanced antigen presentation profile. So what we see here is that when we compare here in ALS in yellow, healthy controls in blue, what we see here is that this interaction is only happening in the blood of ALS patients. So this interaction, HLA-E, CD94, and KG2C, is unique to ALS blood that is not happening in healthy controls. So that's important because NKG2C, we know that this is an activating receptor in natural killer cells. And this interaction is unique to ALS blood, is unique to NK2 cells obtained from the ALS blood. Importantly, DAP12 is coupled with this receptor, and this is the second most significant deregulated gene in NK2 cells from our data set. And finally, it is important to note that HLA-E is presenting peptides through this interaction, and that's important because we may sequence the peptides that are being presented by HLA-E using immunopeptidomics, which will give us any different clues about the involvement of these peptides in ALS, and that might be important in the future. So we next validated the alterations in cell type proportions using flow cytometry. So now we are using an bias technology, and so we after determining that some cell subtypes were upregulated in ALS, we next validated whether these cell subtypes are indeed upregulated using another technique, which is flow cytometry. So we use CD3, CD56, CD16, and FCER1G, which is a characteristic marker of NK2 cells, to differentiate these cells from the major population of CD56 deemed natural killer cells. So as you see here, this is a summary of the approach. Both natural killer cells and monocytes are CD3 negative. So then we use CD56 and CD16 to differentiate these natural killer cells and classical monocytes with CD16. And finally, we infer the proportion of MK2 cells using FCER1G. So using flow cytometry, we were able also to validate our findings. So again, the regulation of CD56 deemed natural killer cells, NK2 cells, bright natural killer cells, and classical or CD14 monocytes was validated using flow cytometry. And finally, we sought to explore whether the plasma neurofilament concentration as a surrogate biomarker of neurodegeneration could be predicted not only by the proportion of our most interesting cell types, so NK2 cells, bright natural killer cells, and CD14 monocytes, but also with gender, age at onset, age, disease duration, age at blood collection, ALS functional rating scale, so different clinical demographic variables, but also the proportion of cell types. So the combination of this approach, of these variables using a regression model, give us that CD56 bright natural killer cells together with ALS functional rating scale, disease duration, and gender are able to predict or to explain almost 65% of the variance on in neurofilament concentrations. We next validated or replicated the result using the proportions obtained from the same cell subtypes using flow cytometry. And in this case, using the proportion obtained from flow cytometry, we obtained a better model, in fact. So giving us the idea that this combination, the combination of these inputs may explain almost 77% of the variance in neurofilament light chain concentration, so potentially explaining neurofilament from a non-invasive biofluid. So we knew that's not novel, so we knew that natural killer cells are increased in ALS, but as I mentioned before, these approaches also, these approaches used flow cytometry, so they were focused on CD56 dim and bright natural killer cells and not in the subpopulation of these cells. We knew that natural killer cells infiltrate the brain. They in fact regulate the microglial phenotype, they regulate neurodegeneration, and they even block T regulatory cells, which are supposed to be good to reduce neuroinflammation. By the other hand, we know that natural killer cell regulate T cell responses, and in fact, they limit the expansion of CD8 positive T cells using the FCER1 gamma, the main marker of our NK2 cell population, but also the gene which is the most upregulated gene in NK2 cells. So this is pointing towards, again, the limitation of the proliferation of CD8 positive T cells by NK2 cells. And finally, different studies report controversial results. So some of them report that natural killer cells in the brain are good, so they clear alpha-synuclein, and their depletion, in fact, exacerbates alpha-synuclein deposition. By the other hand, they might be detrimental. So this in this aging model in humans, they saw that the accumulation of natural killer cells impairs cognition and neurogenesis. So the question here may be, are they good? Are they bad? Does it depend on the stage of the disease and disease progression? And does it depend on the natural killer subpopulation? So we may have, we need to study the whole landscape of populations, not just dim or bright natural killer cells. This is a question that we don't know how to answer right now, but our summary says that natural killer 2 cells are important and the most altered cell type in our study. So I hope I have convinced you that the peripheral immune system has a role in ALS and other neurodegenerative diseases, but in this case, natural killer 2 cells, a subpopulation of CD56 dim natural killer cells, is the most altered cell type in ALS, but also CD14 monocytes and bright natural killer cells. Importantly, so gene expression alterations are found in these cell subtypes, that the MHC1 pathway is in the interaction within the MHC1 pathway is increased in NK2 cells, and in fact, this ligand interaction, this ligand receptor interaction is unique to ALS blood. And finally, that the proportion of CD56 bright natural killer cells, which I did not mention this before, but they have an increased potential to infiltrate into tissues as compared to the dim counterpart. So the proportion of these bright natural killer cells, together with other clinical and demographic variables, is able to explain almost 77% of the neurodegeneration profile in the blood of ALS patients. So a limitation is that this group of patients, this group of 14 patients, did not carry any disease-causing mutation. They were sporadic. We rule out all known mutations. So maybe other genetic forms of ALS, maybe C9 repeat expansion carriers, tardive mutation carriers, TBQ1, or even SOD1, which is not associated with TDP-43 or FAS, may have a different immune profile in the blood. Importantly also is that maybe during disease progression, this profile may change. So it will be important to evaluate whether these cell types are increased when disease progresses. And finally is the thing I mentioned at the beginning of the talk, that peripheral blood mononuclear cells, when you isolate peripheral blood mononuclear cells, you do not include the neutrophils, basophils, or eosinophils. The important point here is that neutrophils have been already associated with other diseases, and that might be a limitation because we cannot isolate neutrophils using the fecal gradient centrifugation approach. So in conclusion, natural killer cells play a major role in ALS. That this is not new, but the point here is that NK2 cells are key players in controlling the peripheral immune response in the blood of ALS by the interactions between using the MHC1 pathway with other T lymphocytes, because we have found like almost 40 genes deregulated in these cells, and they are driving the increase of natural killer dim CD56 dim natural killer cells in ALS, but also the proportion the proportion of bright natural killer cells might parallel neurodegeneration, enhancing the prediction of neurodegeneration from accessible biofluid. And this is also important, as I mentioned before, are natural killer cells good, but well, does it depend on the population? I think so. So NK cell-based therapeutics should consider the whole landscape of natural killer population. So we cannot block the entire population of natural killer cells, because this may have important consequences in the immune system of the patient or the participant, but also that we don't know if blocking the whole population of natural killer cells will be beneficial. Maybe the one that we may block is the bright or the NK2 population. We don't know, but taking into account the whole population of natural killer cells and blocking it, I think is not the best way to impact on the modulation and the progression of ALS. And finally, characterizing the landscape of HLA-E presented peptides, but also modulating this ligand receptor interaction in ALS may be novel therapeutic strategies in this disease. And finally, as the next steps in this related to this study, so we have now single-cell RNA sequencing from the same 14 ALS patients obtained 1 year after this single-cell RNA sequencing approach. So we have now the longitudinal profile of the same samples using also single-cell RNA sequencing. We are now isolating natural killer-derived extracellular vesicles to characterize them not only in ALS, but also in other neurodegenerative diseases, and investigate the impact of these exosomes and whether they could be used in therapeutic approaches. We also are now assessing the impact of NK2 cells with motor neurons derived from iPSC cells. And finally, this is not a funded project yet, we want to characterize the landscape of HLA-E bound peptides using immunopeptidomics in ALS and other neurodegenerative diseases. And by the other hand, other projects involving the immune system, also in ALS, in this case, is the one funded this summer by Ayudas Unzué-Luzón, which will characterize the CSF immune cells using single-cell RNA sequencing. This is one of the major points here. We are investigating the blood, what happens in the CSF. Are these cells in the CSF? Yes, they are, but what's happening there? We also have other projects funded to investigate the immune system at single-cell resolution, in this case in people with Down syndrome at different stages of the Alzheimer's disease continuum. So not only in Down syndrome people with Alzheimer's disease, but also in preclinical stages and stratified by A-beta and tau positivity and negativity. And finally, we have a project to investigate the peripheral blood immune system using single-cell RNA sequencing in frontotemporal dementia. So that's it. Well, I want to thank the participants and healthy controls to participate in this study. Funding agencies, especially Fundela, for funding this work, and our small team of highly talented people, Laia, Kim, Natalia, and Esther, and that's it. Thank you.
Questions? I'm sure we have a lot of questions. Minerva, I see you writing. You told him. Okay. You don't have more questions. Okay, I have. I mean, Oriol, you know the work we are doing with Minerva, trying to. I mean, you were part of the paper of Anya Schneider. So, what do you think about these TDP-43 inside the vesicles? Where do they come from? Does it come from the brain, from the inflammatory cells of the brain, peripheral? What is your what is your hypothesis?
Maybe. Ay, gracias. Me está ahogando todo el rato, perdón. Tengo una tos aquí.
I guess you discussed that before, right?
No, so I don't know whether they come from the brain or not. What we know is that 99% of the cells do not come from the brain, so they are peripheral vesicles, so maybe coming from the kidney, the liver, the blood. I'm not sure. What we know is that phospho-TDP-43 is aggregated also in the dermis, and we found these aggregates in other places beyond the brain. So maybe the cells around these aggregates in other tissues beyond the brain may explain that.
You know, I was talking to you before that we also find this signal in late. So that that could be also a disease in which peripheral inflammation could play a role.
Late and later?
Yeah.
Yes, I think so. I think all neurodegenerative diseases are related to, well, the neuroinflammation process is also, so is not only microglia and astrocytes, is peripheral immune cells contribute to this process. So I think so.
So what do you think is the role that the peripheral inflammation is playing here? Is promoting what is happening in the brain, or the brain is recruiting these peripheral cells to participate in the party?
Yeah, this is a very, very important question. So we know that peripheral immune cells are recruited to the brain using the blood-brain barrier, the blood-CSF barrier, but also the skull channels. We have some channels in the skull, and there are a huge, there is a huge number of immune cells there, a huge reservoir. And well, we also know that the immune cells in the brain, microglia, astrocytes, they present antigens. They are the borders, um, and that the leptomeninges are completely populated by immune cells, the dura mater, in fact. In my opinion, there are two ways. So, of course, peripheral immune cells are infiltrating into the brain, but also the cells which are at the brain borders have something to do with this process. And I don't know if they are good or bad. Some studies they say that they are good because they clear alpha-synuclein. Some others say that they are bad because they impair neurogenesis, and I don't know.
One of the proteins that was expressed in NK2, it was TYRO-BP, right? Which is the receptor of TREM2, right? The microglia receptor. So they share a common pathway with microglia. So.
Yes. Yes. That's really interesting.
Yeah.
This is really upregulated 10 to the minus 63.
Yeah.
And yeah, I don't know why.
Yeah.
But it's expressed in natural killer cells and upregulated in NK2 cells.
Yeah.
Yeah, thanks. I think I have a bit more of a methodological question, and that is, if I understand correctly, your classification of different cell types based on the RNA-seq data is, well, basically based on it's a clustering approach based on the cellular content, well, of the RNA-seq results, right? So, but then, and there's some heterogeneity in this classification, so that you don't have on spot that cell type has that signature, but they're like, these are like blobs, clusters, and they can be quite close to each other, right? So having that in mind, and then you formulate, okay, that's a different cluster, that's the major populations, and that's why you talked about different levels, level 1, 2, 3, because they're within these major populations, you can distinguish smaller subclusters, but then they get less and less different from each other, right? Because the clusters, since they're not really, that there's some heterogeneity within each cluster, they can get quite close to each other, right? So now we know or can assume that ALS or any other disease alters the RNA-seq results, right? So different RNA content basically in the cells. So then the classification of these cells. So how can you know that it's less of these or more of these cells, and not just it's a different cell type that has been misclassified as that cell?
Yeah, I understand the question. Okay. Yeah. So the point here is that so we have the RNA content within each cell, but some of the RNAs are like markers, are genes that are always highly expressed in these cells. So for NK2, FCR1G is a marker, but also, can we share the presentation again? So I will show you a slide that I didn't show before. So the point here is that we have some markers that are not supposed to be deregulated, so are always highly expressed. So they do not depend on the alterations of gene expression. That was the question, no? So if TYRO-PP is differentially expressed, is this another cell type? Or, yeah, so.
Exactly. So the question basically is, how can you know there are, let's say, less cells of one cell type and not just they are different. These are still the same cell types, but they behave differently, and that's why they have different.
Well, it's more complex that the thing I'm explaining right now, because you can see here that is the granularity. So as you hear, no? So NK2 cells, NK1, and how do you know which is the?
So maybe that maybe more of the
Yeah.
NK1 cells have been classified as NK2 in ALS, and that's why
Yeah.
that's
So at
basically
some
my
point,
question.
nothing is perfect. But the other thing is that, of course, there are transitional states, which they do not belong to NK2 or to NK1, so they are transitioning from one cell type to another. These are cell states. So depending on, in fact, the slide that I want to show you is the, let me show you. So NK bright cells are the ones which are represent 10% of the NK cells here. So they express high levels of C56, and the other cell type which express cell type C56 at medium levels is NK4. So not just based on that, but also based on the expression of these markers, XCL1 and 2 and granzyma key. We know that NK4 is transitioning, so NK4 is a transition state between bright cells and NK3 cells. But the point again here is that the markers are never changing. In ALS or healthy controls, they are classified together. So we have this group of cells and we decide that this is a marker, and then we decide to split them in healthy controls and ALS. So we have a marker which is SPON2, NKG7, or granzyma B for NK2 cells, and these markers are always highly expressed by NK2 cells from healthy controls, and always highly expressed by NK2 cells from ALS patients. So you never are influenced by differential gene expression changes in these genes because they are selected as markers, that's the best markers to differentiate NK2 cells from NK1, NK4, NK3, and bright.
Yeah,
See?
absolutely. Answered my question.
Hi. So the question is, what the question by Pascual is there is an autoimmune component, or is that a protective mechanism? So you started with very nice histological images. Yeah. Wouldn't be once you have identified the population, wouldn't it be logic to go back to the histological level and to see where are these cells, what are they doing?
Yeah. Yeah, we have been fighting against natural killer cells and the brains because we are not able to detect natural killer cells easily. So we use NKp46 as a marker.
Yeah.
And what we don't understand is why this marker is highly expressed by some brains, but is not expressed in other brains.
Mm.
And as far as I have read, postmortem interval and of course the different methodologies you use when you stain natural killer cells are influencing your detection. So natural killer cells are like more difficult to stain than microglia, for instance. So when we use FCR1G, which is highly expressed by microglia, in fact, we see microglia and some cells that look like natural killer cells because they are not this ramified structure of microglia. They are round cells and with no ramifications. So we know that these are not microglia. We infer that these are natural killer cells, but when we use the same within the same immunofluorescence, we use NKp46. NKp46 does not stain this cell type. So yes, our next step, of course, is trying to stain natural killer cells in the brain of ALS patients, but till now, we have not been able to find the best protocol.
Okay, but yeah, I understand that, of course. But if you could identify them through single cell sequencing on brain tissue.
Using single nucleus RNA sequencing then. So this is another major problem. So single nucleus RNA sequencing, you know that you are sequencing the nucleus. Then when you isolate the nucleus, so this is not a fresh sample, this is a frozen, normally, it's frozen brain tissue. So then you isolate the nuclei, and then this process is highly biased towards the biggest nuclei, and you know, the most common population. So it is biased towards neurons, inhibitory, excitatory, but also to microglia, astrocytes. And then if you read the studies have used single nucleus RNA sequencing, you see that microglia, they also identify one population, and we know that this is not true. So you can just think about that on whether you can detect natural killer cells using single nucleus RNA sequencing. I think it will be very, very difficult. Some studies, some recent studies have detected T cells, but natural killer cells, which are less common, I'm not sure that they will be detected.
Okay, thank you.
Maybe using spatial transcriptomics. This is
Yeah.
the approach.
Mm.
Well, it's expensive, but spatial transcriptomic
Yeah.
will really maintain the structure of the brain. You don't need to isolate. You don't have to isolate the nuclei, so you have the real structure. You conserve the environment and the cells where they are. So maybe.
Yeah, maybe the pre-analytic data factors of the tissue may influence that also. So we have very, very good tissue for that kind of studies.
Thank you.
More questions? Last one. You mentioned the genetic cases. Are there studies in genetic cases, genetic ALS, and is the same mechanism there?
I found a study published like 3 or 4 years ago, but of senataxin mutation carriers, a very, very, very rare disease caused by a gene related to ALS, senataxin. And they found CD8 positive T cells or CD4, not natural killer cells. No, not all genetic forms. I don't have any information.
Give you a clue about the origin of
Yeah. Yeah, in fact, we know that progranulin mutation carriers and C9 repeat expression carriers are linked to autoimmune disease. So, well, maybe they have a different profile, immune profile, than sporadic patients.
Okay. Well, thank you very much. That's great discussion.